Dr. Choi.
Thank you very much. My name is Hoon Choi. I'm working at Massachusetts General Hospital in Harvard Medical School. And the title of my project is Impact of Exercise Hormone Irisin on Tauopathy. Irisin is a hormone that we produce while we are doing exercise. And I'm studying whether irisin reduce tau pathology, and if so, the mechanism. So it is well known that exercise decreases the incidence of Alzheimer's disease, but we don't know how this occurs, and we don't know how to take advantage of it therapeutically. So one thing we know is that exercise increases BDNF. BDNF is brain-derived neurotrophic factor. It's a protein produced in the brain, and it plays very important roles in diverse brain functions, such as learning and memory. Several years ago, we found that increasing BDNF and neurogenesis. Neurogenesis is a number of stem cells. If we increasing neurogenesis and BDNF, mimic the exercise effect on cognitive function in Alzheimer's disease mouse model. So basically what we found was that if we increase neurogenesis, the number of stem cells and BDNF, this mouse showed the improved cognitive function without exercise. However, we don't know how exercise increase BDNF in the brain. Our collaborators, Dr. Bruce Spiegelman and Dr. Christian Wang, some years ago, they found that exercise increased the secretion of hormone called irisin from the muscles. And this irisin moves to the brain, and it causes the BDNF elevation. So this slide just shows you what the tau pathology is. This is a brief introduction. So in the brain, we have a cells called neurons. Neuron looks like this, and each neurons have a cell component that is called microtubule. The microtubule provides structure and shape of neurons. So it's like imagine like a bones in our bodies. And there's a small proteins called tau. Here is the yellow proteins. These tau proteins, stabilize microtubules. However, in AD brains, what happened is these tau proteins become pathogenic. We call pathogenic tau, or in science terminology, we call hyperphosphorylated tau, p-tau. And the p-tau, pathogenic tau, what happens is they do not support microtubules. Then what happens is the microtubules disintegrate and neurons die. So imagine we don't have bones in our bodies, then we may die. And this p-tau, pathogenic tau, stick together, and they make structures called neurofibrillary tangles, which is very toxic in AD brain. This figure just shows that tau tangles in real Alzheimer's brain tissues. And all these papers, not limit, I mean, there are more papers outside, but these papers show that exercise reduces tau pathology in various mice. However, we didn't know, I mean, so we asked whether irisin, exercise hormone irisin, is responsible for the exercise effect on tau pathology. So in 2014, we made the three-dimensional human neural cell culture models of Alzheimer's disease, like here. This is like a brain in a dishes. So we treat the irisin in the 3D cell cultures and see and study what happens. So then we found that irisin reduced tau pathology in 3D cultures.
Oh my God.
Okay, so here, the dark band represents the amount of p-tau in the cell culture systems. Here, 0 means we didn't treat the irisin in these cell cultures. This is like we say control. And 5 means we treat the low dose of irisin in the cell culture system. 500 means represent we treat the high dose of irisin in our 3D cell culture system. And you can see if you compare black band here in the red boxes compared to black band in yellow boxes, you can see irisin reduced the amount of p-tau in our 3D cell culture system. And also we took a picture of the neurons. Here, this is the picture. And the blue cells represent the cell neurons that contains p-tau. So you can see when we treat the irisin, 3D culture produce fewer numbers of p-tau positive cells, blue cells. So all these results, prelim results suggest that irisin could reduce tau pathology in our 3D cell culture system. So based on this result, our prelim result, we hypothesized that irisin has neuroprotective effect in Alzheimer's disease by reducing tau pathology. We decided to check this idea, whether it's true or not, in two cell cultures, in two systems. One is we'll test our idea in cell cultures in aim1. In aim2, we will test whether irisin reduces tau pathology in mice. So in aim1, we'll test whether irisin reduces tau pathology in cell culture models. So basically, we'll treat irisin in the cell cultures, this in vitro cell culture system, and we'll determine whether irisin reduces tau pathology. And if so, we'll find the mechanism by which irisin reduces tau pathology in our 3D cell culture system. For example, there's a protein called tau kinases. Tau kinases is a protein which make a pathogenic tau. So these kinases, tau kinases, convert normal tau to pathogenic p-tau. So we'll test whether irisin treatment to weaken the activity of tau kinases so that the neurons doesn't make enough lots of p-tau proteins. In aim2, we'll test whether irisin reduces tau pathology in mice. So basically, we'll inject irisin into the mice. We will use tau transgenic P31 as mice, and we'll determine whether irisin reduces tau pathology. And we'll test whether the identified mechanism from the cell culture system in aim1 is validated in mice. We'll also explore whether irisin improve cognitive function in these mice. And also we'll test whether irisin treatment slow down neurodegeneration and or neuroinflammation. Of course, we'll also test whether irisin injection is safe in animals. So year one, we'll complete the cell culture system as I mentioned, and we'll prepare for mouse experiment. We start breeding and aging P31 as mice. We're also starting the injecting irisin in these mice. And year two, we are planning to complete the animal experiment in year two. So for long-term experiment, our long-term goal is we'll examine irisin effect in humans. So this is our expectation. We expect that our work will establish a framework of how irisin could be used as a therapeutic target for Alzheimer's disease. Also, our work will provide a better understanding of molecular mechanism, how exercise reduce tau pathology in Alzheimer's disease. So they are my collaborators, Dr. Rudy Tenzi. I'm collaborating with Dr. Rudy Tenzi. He was my postdoc mentor. I'm still working in his group. And I'm working with Dr. Bruce Bigerman, who found irisin for the first time. And I'm working with Christian Wan, who found that irisin increased BDNF in the brain. And we all greatly appreciate for CART for the generous support for our research.
Thank you for a fascinating presentation. And so we have a check to support your work from Bill Parker and myself for $100,000. Congratulations.
Thank you very much.